Welcome back to part 2 of chapter 6 on metabolism. So before we move on to the next section of the chapter, let's look at these 4 examples and see if you can tell me whether it is an exergonic or endergonic reaction. Think about the keywords I'm asking for when we're going through each image, and think about in terms of enthalpy and entropy value and what's being changed in the certain scenario to think about whether it's exergonic or endergonic in terms of what we just went over in part 1. Okay, so for the image right here, image A, you have a pile of compost in this area right here, which, as we all know, composts are really breaking down organic matter into smaller minerals that can be reused as fertilizers for plants. So is this an exergonic or endergonic reaction? Now think about the keyword. We're breaking down organic matter into smaller minerals. So is the entropy value increase or decrease? It's a higher entropy value, okay? And in terms of enthalpy value, we are actually decreasing the enthalpy value because we're going from bigger organic matter into smaller minerals, okay? So hence this tells us that this is an exergonic reaction, okay, with delta G being negative for this compost breaking down reaction. Okay, now on to image B here. You have a chicken embryo forming within the egg, okay? When a chicken is being, formed in the egg, all those cells, tissues, and organs will have to come together to form a more complex organism, okay? Is this an exergonic or an endergonic reaction? Now think about what is happening to entropy value. We're going from random little cells and tissues, and we're forming in them into an organism, okay? So we are decreasing entropy value, okay? We are making a more organized organism, and hence our enthalpy value is increased because we're really storing all those going from a lower energy value of those random stuff and putting them into a more complex organism, okay? So it's increase in enthalpy, okay, which is why the formation of an embryo still requires energy. Okay, that's why, in terms of a chicken, they need the yolk there to provide all the nutrient and energy to power all those reactions that's for forming the embryo into a baby chicken. So in the end, this is an endergonic reaction, and delta G is positive value. Okay, now on to image C here. So in image C, you have a very neat pattern of sand art to begin with on the sand, as you can see by these little lines and spirals and stuff. Let's say someone decides I'm going to throw a ball into that nice pattern and let the ball roll all over the place. Now you've just messed up that whole nice sand art pattern that was formed on the sand. Is this an exergonic reaction or an endergonic reaction? Now, easy way to think about it is this getting more messy or is this getting more ordered? It's getting more messy. So increase is in entropy value makes it an exergonic reaction. Okay? All right, and in the last image here, in image D, we have a ball rolling down the hill all on its own, starting from the top of the hill to the bottom of the hill, okay? So we can think about this in terms of turning potential energy into kinetic energy, okay? Potential energy at the top of the hill, and then kinetic energy that enables the ball to roll down the hill. So is this an exergonic reaction or endergonic reaction? It's an exergonic reaction because it doesn't need any extra energy for this to happen. There's already potential energy at the top of the hill, and we're just using that in forms of, and turn that into kinetic energy in order for the ball to roll down the hill. Okay, we're changing from a higher energy to a lower energy value. Okay, we're decreasing the amount of enthalpy, and that's why it's an exergonic reaction. Okay. All right, so now that we understand the concept of energy and whether we're releasing energy or requiring an input of energy for a chemical reaction, we can go on to our next topic of interest, which is the currency of energy in form of ATP, adenosine triphosphate. We were talking about earlier how photosynthesis is an endergonic reaction and that it requires an input of energy from the sun in form of light energy. Well, but in our human cells, we have a lot of chemical reaction that require energy to get started as well, but yet we don't have chloroplasts or chlorophylls, okay? And chlorophyll is what make plants green, and that's why we humans are not green-skinned. We can't go out into the sun and go and then we're getting energy from the sun. We don't need to eat. That is not the case, right? So we cannot absorb sunlight as a source of energy. That's just what we humans don't do. So what we do have, though, instead, as our form of input of energy for all the endergonic reaction that has to happen inside our cell is ATP. Okay, the molecule contains the energy we need to perform the work of any chemical reactions in our cells, just like how we need money to buy something we want. For example, a coffee we want to drink or a sandwich we want to buy for our food. Our cells need to have some way to quote unquote buy the products that we want. If a cell needs to move things from one side to another, sometimes it's not a spontaneous process. It might require the cell to buy quote unquote a particular enzyme to move those things. Or if we go back to the story of lifting weights in the gym, we want to move our muscles, but it is not a spontaneous process. Our muscles can't just move on their own. We need to pay our muscles in form of ATP in order for them to produce a movement. Okay, now I guess that's where the pun comes from. The saying goes, there's no such thing as free lunches. Just a way to think about it. Okay, but we have to pay our muscles in form of ATP in order for them to move, and that payment, like I said, is ATP. Okay, it is through this high energy molecule that we are able to power all cellular reactions. Okay, now as we have discussed in chapter 3 for nucleic acids, there were three main components. Remember, in the nucleotide, okay, we have the nitrogenous base, the ribose, which is the 5-carbon sugar, okay, pentose, and then we have the phosphate group. That was the three basic component of a nucleotide. Well, ATP is actually very similar to the general makeup of nucleotides. It still has a nitrogenous base right here, but now it is, since it's ATP, there's a reason why it's called adenosine, is because it is only made up of the nitrogenous base adenine. Okay, we don't have thymine, cytosine, or guanine. Okay, ATP is made up of adenine, as is nitrogenous base. It also has the same 5-carbon sugar ribose right here as we have seen. And then lastly, in the third component of ATP, instead of having just one phosphate group, and that's why the name triphosphate, it has 3 phosphate groups at the end right here. 1, 2, and 3. Okay, 3 phosphate group that makes up ATP. Okay, so it is those bonds right here that store a lot of energy, and that is why ATP can serve as a high energy molecule that can provide energy for many of our cellular processes. So, how do we spend our ATP to release energy in our cells? Now, to release energy, ATP has to go through a chemical reaction we learned about in chapter 3 called hydrolysis. And in this case, specifically, ATP hydrolysis. Okay, so ATP hydrolysis. And that means, remember, hydrolysis is for breaking bond. Okay, so we're breaking that ATP bond between the terminal, this is the terminal phosphate, and this is a second phosphate. This right here is that high energy bond we're going to have to break in order to release energy. Now, technically speaking, the bond right here and the bond right here is pretty high energy too, but it's just not as high energy as this one right here, which is why usually for ATP we only break this bond and then we remake this bond to regenerate ATP. Okay, all right, so with that being the highest amount of energy, when a phosphate group is broken away from ATP, in turn it turns into what we call an ADP molecule, okay, which stands for adenosine diphosphate, with only 2 phosphate group, because one of those phosphate group has broken off, and we call that our inorganic phosphates. Okay, so inorganic phosphate group. Okay, and this is considered as an exergonic reaction because it releases energy from the phosphate bonds, okay, of those phosphate group. Now, the exact amount of energy that can be released from ATP hydrolysis, and I've just covered it up. There you go. Let me rewrite that. Okay, so it's ATP hydrolysis, and this reaction itself is exergonic reaction, and this is why you see a negative in front of the delta G value, and we can actually quantify that exactly into negative 7.3 kilocal per mole. Okay, that's our delta G value for the reaction of ATP hydrolysis. That's how much energy we are able to release by breaking that one particular high energy bond of the terminal phosphate. Okay, and hence this energy that is released right here by ATP hydrolysis can be used to power other endergonic processes in the cell. Okay, now don't mistaken when you're studying for your exam, oh, ATP hydrolysis endergonic. Wrong. Okay, what this is saying is that ATP hydrolysis is an exergonic reaction, giving you a negative delta G value, 7.3, and we can use this energy to power other endergonic processes in a cell. Okay, other endergonic processes, not that ATP hydrolysis is an endergonic process. Okay, it can be used to power other endergonic processes. Okay, and also this number right here will be a number you have to remember. Okay, one ATP molecule is always able to generate about 7.3 kilocal per mole, and remember that is negative because that's the energy that is released by breaking one molecule of ATP. Okay? Make sure you remember those things. Now, the exact amount of energy that can be released from ATP hydrolysis was 7.3, like I was saying, and hence, this energy that is released can be used to power, like I said, other endergonic processes in the cell. Now remember, endergonic reaction requires an input of energy, and as I've said, since we humans can't use sunlight as a source of energy, we have to rely on this energy from ATP hydrolysis in order to power those other endergonic reactions. Okay, we can also make ATP molecules through dehydration reaction. Okay, dehydration reaction. And again, something we talked about in chapter 3, dehydration is where a bond can be formed. And in this particular case, we're forming a bond again from ADP molecule and an inorganic phosphate between these two things to remake ATP once again. Okay, so if I was to put this in a reaction form, ATP hydrolysis is ATP breaking down into ADP plus inorganic phosphate. Okay, ADP. And the amount of energy for this reaction is negative 7.3 kilocal per mole. Okay, and ATP dehydration reaction is ADP plus inorganic phosphate will turn back into your molecule of ATP, and this reaction is an endergonic reaction which requires the exact same amount, 7.3 kilocal per mole of energy in order for us to make this molecule of ATP. Okay, so this is an endergonic reaction. This is an exergonic reaction. All right. Okay, so this is technically, like I said, dehydration is an endergonic reaction. Okay, we will require an input of energy from other exergonic cellular reaction. You see, energy coming from other exergonic cellular reaction. Okay, that does not mean that dehydration is an exergonic reaction. Dehydration is a positive value, so it's an endergonic reaction. But in order to power this formation of ATP again, we need exergonic reaction from other reaction in the cell in order to provide us with enough energy to remake ATP molecules. And which is what we will be talking about when we get to chapter 7 on cellular respiration. How do we make ATP? What enzymes are involved? Where do those input of energy comes from in a cell? We will save that for chapter 7 to discuss. Okay, for this, we just need to know what is ATP hydrolysis, what is ATP dehydration, what is the value of delta G for both reactions, and what does that mean for, is it providing energy for other processes, or is it acquiring energy from other processes in the cell? So what we have just discussed with ATP hydrolysis and ATP dehydration reaction is a form of what we call energy coupling, where free energy of a reaction is functionally linked to energy needs of another chemical reaction. The amount of energy that we make is used to power another reaction somewhere else. That's what we mean by energy coupling. So for example, there is a very important protein we will talk about in anatomy and physiology when we learn about how our body is able to reduce a muscle contraction via an electrical impulse coming from a neuron. Notice how this particular protein is called sodium-potassium pump, and it is the perfect example of what we mean by energy coupling. So the sodium-potassium pump in a cell is in charge of driving sodium ions out of the cell and potassium ions into a cell. And typically speaking, this is happening against its concentration gradient. We'll talk about diffusion, concentration gradient, all that stuff when we get to chapter 5, okay? But just know that for now, when we're talking about going against its gradient, it requires an input of energy, which is coming from ATP. So the main goal of sodium-potassium pump is to stabilize cellular concentration of these two ions, sodium and potassium ion. But this is not a spontaneous process like I was saying. It will require an input of energy from an ATP molecule. So this pump is a protein that looks something like this right here, okay, this purple glob right here, and it sits in the plasma membrane. So this right here is really representing your plasma membrane. Okay, remember when I talked about phospholipids in chapter 3 and how it makes up the lipid bilayer of the plasma membrane, I mentioned that a cell's plasma membrane is actually not bare with nothing on it but phospholipids, but it is actually filled with things bobbing in and out and going through the plasma membrane. And this is one of those proteins that goes through the plasma membrane to serve as some kind of pump for these two particular ions, sodium ion, potassium ion. Okay, and what we have here is represented by this is what's outside of the cell, and this is what's inside of the cell in the cytoplasm. So this particular protein is very special, where it is divided into two sides, one side here and one side here. Okay, and these two sides are very structurally different from each other with different pockets for different ions. This side of the proteins has specific pockets for sodium ions, and that's why it's, you know, in this circular shape to fit those circular sodium ions. The other side right here will become specific for potassium ions. But for now, there is no potassium ions binding to it because this particular pockets has not been changed yet. The protein, this whole purple glob here, actually has to change in structural conformation in order for it to reverses which ion it binds to. So currently in this particular conformation, it is only binding to sodium ions. And once sodium ions are bound, a molecule of ATP can actually be hydrolyzed to ADP and inorganic phosphate, and the inorganic phosphate group actually gets transferred from the ATP onto the protein pump itself. Okay, it becomes attached to the protein pump. Okay, or the sodium-potassium pump specifically. And what happens is when it gets attached, we call this process phosphorylation. Okay, you will see this word a lot in upper level biology classes when we talk about enzymes and proteins in a reaction, because phosphorylation is something that will be mentioned quite often as a way to either activate or inactivate an enzyme or protein. So the energy that is released from this ATP hydrolysis right here, okay, will power the change of in conformation of the sodium-potassium pump and turns it upside down. Okay, well, not really upside down, but opening. It turns it to open up on the other side towards the outside of the cell like this. Okay, kind of like a little Pac-Man thing, and I'll show you a video in a bit about this and you will have a better idea what I'm talking about. And once this sodium-potassium pump conformation has been changed to be the other way around, it dumps the sodium ions to the outside of the cell. Now with this conformation change, you see the differences in the shape of these pockets right here. Okay, from this circular shape of these pockets now has become smaller to kick out the sodium ions. The other side here now becomes more open, as you can see right here, more open compared to before. So now what happens is potassium ions can now bind to the sodium-potassium pump to fit perfectly into that little pocket or notch on this side of the protein molecule. Okay, now so you can see the differences in the shapes of these pockets that allows things which ion to bind and which ion to go out. And once this protein is bound with potassium ion, it triggers another conformational change that will cause this terminal phosphate group to be released from the pump. Okay, so this inorganic phosphate comes right off. Okay, and that terminal phosphate coming off of the molecule will cause and trigger a change in the protein molecule. Now back to this way around, okay? Opening up towards the inside of the cell, back like the beginning, okay? So once the phosphate group is no longer there, okay, it will revert back to its original position, as you can see, and now potassium ions will come out of their pockets because that pocket is going to change in shape, and now those thinner pockets for sodium opens back up again, and sodium ions can once again bind to those particular pockets to dump more sodium ions outside, okay? So it continuously go open towards the inside to open towards the outside to dump sodium ions out of the cell, take potassium ions from outside of the cell and dump them to the inside of the cell. Okay, again, the animated video I will show in a bit will have a clearer picture of what's going on here if you are confused. So the pump's action in utilizing ATP to pump sodium ions out of the cell and potassium ions into the cell is a perfect example of how energy release from ATP hydrolysis is able to couple with the energy that's required to power the pump and transport these two particular ions. And many other cellular metabolic reactions usually requires molecules or proteins to alter in that slight conformation in order for a reaction to proceed. And oftentimes, like I was saying, it is usually achieved by phosphorylation. ATP hydrolysis to create ADP in an organic phosphate. Inorganic phosphate gets attached to the protein itself. That's what phosphorylation means, to cause a difference in conformational change, which will then drive the reaction to continue on. Okay, so the energy that is released, so for example, we already know that ATP hydrolysis, okay, is able to create energy, okay, and that energy then is used to power this particular pump, okay. So here's the video that we will talk about. Here's that promise animation video that I talk about to show you how the action of sodium-potassium pump with ATP works together in energy coupling.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. 1 phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Okay. So the energy released by ATP hydrolysis usually cannot supply the energy needed by another endergonic reaction, which is what we're talking about in terms of energy coupling. And sometimes it can even provide more energy than necessary, and we can actually quantify how many times can one ATP molecule power the same endergonic reactions over and over and over again, and we know how much energy is needed to run that reaction. So for example, we already know that ATP hydrolysis is able to provide an energy value of negative 7.3 kilocal per mole in terms of delta G. So if an endergonic chemical reaction of turning glutamate and ammonia into glutamine, this reaction is particularly an endergonic reaction and requires a delta G of positive 3.4 kilocal per mole of energy. Is the amount of energy provided by one ATP molecule enough to power the reaction? And what will be the overall delta G of this energy coupling between ATP and the endergonic reaction right here? Or in a sense, is this reaction endergonic or exergonic overall then? So let's go through the calculation. Okay, so we have one molecule of ATP that is able to provide us negative 7.3 kilocal of energy. This reaction right here, okay, requires positive 3.4 kilocal of energy. So we put those together and we find that we have a leftover of 3.9 kilocal of energy, and it's still a negative value, which means, yes, this particular endergonic reaction can be powered by one molecule of ATP hydrolysis, and that we have negative 3.9 kilocal of energy left over. And since this is negative, the overall reaction will still be exergonic, okay, because the reaction is still a negative value. Now, a lot of times, I think students tend to overthink this type of problems because on the exam, all I do is switch to another reaction, but it's still the same concept, okay? You have reaction A here, okay? This is how much energy it requires to run the reaction. This is how much energy you have from one ATP molecule. So is this enough for this reaction to go, okay? It's kind of like back to that idea of currency, okay? You can think of it this way, if this makes it easier for you to understand this kind of problem. You have $7.30 in your pocket right now. That's your ATP. That's your currency in your wallet, okay? You want to buy something that costs $3.40, okay? So is the $7.30 in your wallet enough to buy that particular reaction that costs $3.40? Very simple, right? It is enough. You have $7.30. That's $3.90 more than what it costs to buy that reaction, okay? So you can think of it that way to help you remember how to work these problems, because, a lot of students get, sort of stumped by this kind of problem on the exam when it's really just the same thing. It's just changing the problem around. If you think of it as money, then it's a lot easier, okay? So here's a second example that asks, sort of twist the problem a little bit around, okay? The problem says, if it takes 2.1 kilocal per mole of energy to move one sodium ion across the plasma membrane by your sodium-potassium pump, how many sodium ions can one ATP molecule hydrolysis move, okay? So you can think of it this way again. This particular movement of sodium ion across, if you want to buy one sodium ion, it costs about $2.10, okay, which is basically what the problem is saying. Now, in your wallet, again, you only have one ATP molecule, which means in your pocket, how much do you have? You have 7.3 kilocal per mole, or $7.30 in your wallet. So now what this problem is really saying, how many sodium ions can you buy with 7.3 or $7.30 in your pocket when one sodium ion costs $2.10? Well, very easy, just a basic, division problem. Okay, so 7.3 divided by 2.1, it's approximately 3, okay? We can't do half of an ion or point something of an ion. So it's 3, we have to round it to the nearest number. So 3 sodium ions, okay? Or we can think of it as 2.1 times 3 gives us 6.3 kilocal to move 3 sodium ions across via the sodium-potassium pump. And that is enough paid by our 1 molecule of ATP energy that is produced from ATP hydrolysis. So this 1 ATP molecule is able to power the movement of 3 sodium ions, okay? So again, don't let all these words scare you on the exam, okay? When you come to this kind of problems, it's really just multiplication, addition, subtraction problem, okay? Think of it as you having, if it says 1 molecule of ATP, then it says you have $7.30 in your wallet, okay? If it says you have 2 molecules of ATP, then it's 7.3 times 2, okay? Which is $14.60 in your pocket. Okay, and then depending on what the reaction costs, you will know whether you have enough energy for this reaction to go. Okay, all right. Okay, so we saw in our energy graphs previously that whether it is an endergonic or exergonic reaction, there's always this little hill of energy hump, okay, what I like to call energy hump that we must hop over in order for the chemical reaction to proceed. This explains why we said earlier that just because a reaction is spontaneous with a negative delta G, that doesn't mean it will be instantaneous, because we still have to have enough energy to hop over this particular little hill in order to get to our final products at the end of the reaction. And that energy required for the reaction to proceed and hop over is referred to as activation energy. An easy way to remember this is that it is the energy required to activate a reaction. That's why we call it an activation energy. Now, in the case of exergonic reaction, even if no additional input of energy is required, but some energy is still necessary, for example, like the energies for molecule colliding into each other, for example, is still needed to contort a molecule a certain way to go over this little hill before you can become a product. Okay, so that little bit of energy is what's going to contort that molecule to break or form whatever chemical bond for that release of energy. If it's an exergonic reaction, we're breaking a chemical bond for release of energy. If it's an endergonic reaction, we're contorting that bond so that it can be formed and that we can use the input of energy to form the bond in the resulting product. Okay, so either case, we're putting molecules in a certain way in order for the reaction to proceed to products. Okay, again, when you go into chemistry and thermodynamics, you're gonna get into a lot more of this, but this is the gist of what we need to know in biology. Okay, and whether it's an exergonic or endergonic reaction, activation energy is always a positive value. Okay, you will never see an upside down hill. Okay, in thermodynamics from going from reactant to product. I see some students, I used to, in an in-person class, I actually require my students to draw these graphs. So some students do this, okay? That means your activation energy was 0 or something, or a negative value in order to go from reactants to products. That never happens, okay? Activation of energy is always positive, whether it be a big positive value or a smaller positive value. Both of these is an activation energy of some sort, and they are always positive. Okay, you always need some energy for the reaction to proceed. Okay, so an example of that source of energy in a chemical reaction can be the heat energy that is absorbed from the surrounding environment. Heat energy, okay? So the more heat that is applied to the reactants, the faster and with more force are those molecules of the reactant moving and colliding against each other, eventually pushing that molecule over the hill and over our activation energy to reach our final product. This explains why sometimes when we heat up something, it makes a reaction go a lot faster. Is because of that higher frequency of molecules bombarding against each other and more force they're hitting each other with. That's what drives a reaction, in a sense, overcoming that activation energy to get over to the products. And hence, the higher the activation energy value is, the bigger that hill the reaction has to hop over. Okay, and because of a bigger hill, that reaction will be a slower reaction. Okay, the bigger this hill has to be from reactants to the top of the hill, the bigger that hill is, the slower the reaction will be. Okay. And most cellular reaction actually have activation energy that are too high for just heat energy alone to overcome at efficient rates. Okay, that's why I say technically speaking, burning of glucose is a spontaneous reaction, but we may have to wait until we starve to death before it's going over the hill by itself. Okay, it will take a long time. And that's why we require the help of enzymes, okay? Catalysis of enzymes, because enzymes will lower that activation energy hill by a great amount, such that the reaction then becomes a much faster reaction compared to when it was without an enzyme, okay? So here's the last major topic of this chapter: introduction of enzymes and their application in cellular chemical reactions. Now, almost all enzymes are made up of proteins, and if you remember from what we learned in chapter 3 about proteins can denature at certain temperatures, pH, and exposure to other chemicals, which will be something that we will discuss on how these things can also affect how efficient an enzyme can work for a reaction. How an enzyme is able to catalyze or make a chemical reaction go faster. The main thing it's doing, okay, remember to highlight this, note this in your notes, is lower the activation energy of chemical reaction. So for example, in this particular image, let's say this is an endergonic reaction. As we can see, that reactant is lower in energy than your product. That's why it's an endergonic reaction. The original reaction is this purple line right here, going from reactant, a huge hill to climb before you get to your product. So the activation energy always goes from where your energy value is of the reactant to the top of the hill. Okay, so this right here represents the activation of energy without an enzyme. But if you were to add an enzyme to the reaction, you're able to lower that hill by a significant amount right here. Okay, so instead of having to climb all the way up there, when you have an enzyme, we're following the red dotted line right here with a catalyst, which means with an enzyme. And you can climb the smaller hill, and then we're done. And we're going to go over to our products. Okay, so this activation energy right here with an enzyme, okay, is a lot lower than what it would have been with an enzyme. So that's why we say what an enzyme is doing for the reaction is really lowering that activation of energy. Well, then the next question you might ask is, how does it do that? How do enzymes lower activation energy of a reaction? Now, remember we said that heat absorbed from the surrounding is required to contort the reactants a certain way in order for them to break or form bonds between them. Well, if we can have something that can assist in the contortion instead of relying solely on the heat absorbed from the surrounding, then we can make the reaction happen a lot faster. And that's what enzymes are doing. It provides a little pocket just like this. This whole blue thing right here is an enzyme. Okay, remember how we talked about proteins are globular structure. It has all these interactions to give you that three-dimensional shape. Well, here's that three-dimensional shape of this protein, and it turns out to be an enzyme with a little pocket right here. Okay, and this pocket, called an active site, is specific in structure to bind to the reactants perfectly. Let's say if the reactant is glucose, this pocket right here, it's specific for glucose only, nothing else. Okay, if you put sucrose in here, it won't work. Okay, it only works with glucose. So this particular substrate, okay, or reactant, then binds to the active site or that particular pocket in the enzyme, and the enzyme holds them in such a way that makes it more ready to break or form bonds. See how it gets clenched in the middle like that? It then causes those bonds to be more readily broken or bonds more readily formed, and thereby lowering the activation energy for the reaction. Okay, so that's really what enzymes are doing. Okay, it's lowering that activation energy, and how it's able to do that is by contorting or positioning that particular reactant in a certain way within the pocket of this enzyme that makes this reactant more likely and have an easier time to break or form bonds to give you your final products of the reaction. Okay? Now, keep in mind that enzymes can never change the spontaneity nature of the reactions, meaning that an endergonic reaction will always be an endergonic reaction. An exergonic reaction will always be an exergonic reaction. You can never use an enzyme to change an endergonic reaction into an exergonic reaction, nor can you change an exergonic reaction into an endergonic reaction. That is not what the enzymes are doing. Enzymes are just doing the lowering of activation of energy, in this case in an endergonic process. And the other graph here I was showing right here is how the purple line represents the exergonic reaction without an enzyme, and with an enzyme this is how it looks like. Okay, so we're comparing activation of energy from this bag, okay, without an enzyme, to this bag only with an enzyme. Okay, so exergonic reaction with and without an enzyme right here, okay, and then endergonic reaction with and without an enzyme. Okay, you can never change the spontaneity nature of the reaction with an enzyme, okay, because it will never change the reaction's delta G. Remember, delta G is the differences in the energy value of reactants versus product. We're not changing that. Okay, the reactant and product is always in the same line, but what we're changing is this hill right here, okay, which is activation energy. All right, so we usually refer to molecules that bind to a specific enzyme as substrates. And sometimes there can be more than one substrate that will bind to the enzyme. For example, if we're adding substrate A and substrate B together to make a molecule AB, then the enzyme has to bind to both substrate A and substrate B. So depending on the nature of the reaction, different enzymes can do different things. It can bind to one substrate or it can bind to multiple. And different enzymes have, like I said, have the capacity to do a different variety of things. One substrate can break down into multiple products, break down that substrate into multiple products. Multiple substrates can come together to form a larger molecule. Two substrates can enter and then leave as completely different products. And, you can do a whole variety of different things. Now, the location where substrates bind to in an enzyme is called the active site, which is right here. If this was the enzyme, this would then be the substrate, and this right here with these little pockets is the active site. And the active site is very specific in structure such that it fits the substrate just like how a jigsaw puzzle pieces are fitting together. That's how specific things are. That's why I say when an enzyme is specific for glucose, only glucose can bind. You try to turn it into some fructose, for example, it's not going to bind. Fructose will not bind to that particular enzyme. A lot of times enzymes are very specific in what substrate it will bind to, okay? And that's all determined by the three-dimensional structure of the enzyme itself, which goes back to the idea of why I say the structure of the protein means it is the function, is what drives the function of the protein. And because enzymes are proteins essentially, they can become denatured, just as we have discussed with proteins in chapter 3. For example, temperature can change in a reaction if it was exposed to more heat or less heat. Some degree increase in temperature can increase reaction rates. Like we were talking about, the more you add a little bit of heat, you're going to increase the frequency of molecules colliding into each other, or the force as well. But if the temperature becomes so high that it becomes outside of that optimal range of the enzyme, the active site can actually change shape, which is what we call the denaturation of the protein itself. And now what's supposed to be globular in shape to make that little pocket right here for substrate to bind now falls apart into a little string. Okay, well, here's the little substrate that's supposed to bind to that pocket, now has nowhere to bind, can't bind to this little string of stuff. So that's what's happening in denatured enzyme. It becomes such that there's no little pocket or active site for the particular substrate to bind. And too high of temperature can denature enzymes, making the enzyme no longer usable, which is one of the variable you're going to explore in lab 6, okay, and what you're going to write about in your lab report, okay. A second major contributor of enzyme denaturation is the pH value of the solution when the enzyme, whatever solution the enzyme is sitting in. As we have learned already in chapter 3, pH changes can affect the acidic or basic properties of amino acids at the active sites. An extreme pH, either too acidic or too basic, can actually also denature enzyme. Again, going from a very nice 3D globular shape for that particular enzyme to bind it can fall apart completely into a little string where that substrate can no longer bind. Okay, so whether it be temperature or pH, both of those outside of the optimal range of that particular enzyme can lead to denaturation, leading to non-functional enzyme. Okay, all right. Interestingly, though, after we talked about all that, different enzymes may have different optimal pH and temperature conditions. It all depends on where or what the enzyme is working with for its original purpose. Now, for example, pepsin is an enzyme that works in the stomach that will break large proteins into smaller polypeptide chains in order for our stomach to prep our little goop of, liquidy food into the small intestines, and small intestines then further break down your polypeptide chains into individual amino acids, and that's how we are able to absorb amino acids. So pepsin in the stomach needs to start breaking those huge molecules of proteins into their individual chains first. So our stomach, as we know, is actually filled with stomach acid. And I might have already mentioned this in chapter 2 when we're talking about pH, that stomach acid is a super low pH level kind of solution, almost at a pH of 1, that most other proteins will be denatured under that condition. But because this particular enzyme pepsin was designed to work in the acidic environment of the stomach, it actually only becomes activated when it's exposed to such a low pH environment. So that particular enzyme's optimal pH condition is actually very acidic, around pH of 2 even. So on the other hand, a different enzyme called salivary amylase is an enzyme that works in our mouth. It is released in our saliva to assist with the initial breakdown of large carbohydrate molecules that we eat. Let's say we just ate a piece of bread in our mouth, okay? Well, what starts to break down all those carbs within the bread is that our mouth releases saliva. And what's in our saliva is an enzyme called salivary amylase that has a special pocket or active site that binds to carbohydrate specifically. Now, our mouth is not as acidic as the stomach, so the optimal pH for this particular enzyme, salivary amylase, is very close to that of our mouth, which is neutral, around pH of 7. Okay, so or else our mouth is not acidic, or else all our teeth would start becoming not very healthy. Okay, so our mouth is naturally neutral pH, and that's why this particular enzyme works optimally in neutral pHs. So if AMP, anatomy and physiology, is in line with what you have next in your career path or educational pathway, we will talk about these enzymes in detail when we talk about our digestive system. Okay? And along the same line, there are optimal temperatures for different enzymes as well. Now, most of the enzymes that work in our body, of course, has to have an optimal temperature close to our body temperature. And our body temperature is around 37 degrees Celsius. So most of the enzymes that work in our body has an optimal temperature of 37 degrees Celsius. But if we're talking about enzymes in plants or enzymes in some bacteria that lives in hot springs, those enzymes will work at a different optimal temperature. So that's why in your lab 6, one of the things you will explore is figure out what is the optimal temperature of that particular enzyme you're looking at. Okay, depending if it's from an animal or is it from a plant, it may have different optimal temperature than 37 degrees Celsius. And that's what you have to find out in your experimental procedure when you get to lab 6. And what you have to talk about in your lab 6 lab report. The one lab report you have to write this semester. Okay, now once the substrate binds to the active site on the enzyme, it triggers something called an induced fit. What that is basically as the enzyme and substrate come together, the interaction of the enzyme-substrate complex, okay, what we call enzyme-substrate complex, causes a mild shift in the enzyme structure to achieve that ideal fitting to maximize catalysis. If we look at this particular image here, see how this is kind of flat and this is somewhat of a pointy area? Well, this is very circular, there is very curved, and this is very pointy. So when that substrate binds to the enzyme right here, this particular area, although not perfectly fitting to the substrate, will undergo what's called induced fit, so that now it fits and wraps around that substrate very well to achieve the reaction catalysis and release the products at the end. So this induced fit is able to, in a sense, add to the contortion of the substrate molecule that will aid in its bond breaking or forming, depending on the chemical reaction the enzyme is catalyzing. So another thing to add about enzyme is that they remain unchanged by the reactions they catalyze. They are not consumed and release their products once the reaction is completed. Okay? What I'm saying is the enzymes will not change in shape. Just because it has gone through that reaction, it won't become this all of a sudden in the active site, nor would it be completely eliminated. Okay, that doesn't happen to enzyme. They're kind of like little machines in an assembly line, if you can imagine that. Imagine those machines for making a donut, for example. As the donut dough is coming through, okay, that machine is going to come in and drill a hole into the dough to make your little hole in the donut, or chop the donut in half. Okay, so that's all the machine is doing, and then it spits out the final product in either a chopped donut or a donut with a hole. Okay, and then they move on to the next donut dough to go on and chop another donut dough or drill a hole in another donut dough coming through the assembly line. The machine itself are never changed in the process of making that donut, nor is a machine consumed and we have to buy a new machine every time it has processed a donut. So that's what enzyme really is like. Okay, they're like those machines. They're just facilitating the process of making the final product. They're not consumed, nor are they changed. Okay. They can become broken, though, if we give it a wrong condition, just as what we were talking about in terms of pH and temperature. Okay. Now, to add another layer onto our understanding of enzymes and reactions they catalyze, it turns out that we can either inhibit an enzyme or activate an enzyme with other molecules. So let's start by talking about enzyme inhibition. There are two types of enzyme inhibition. One is referred to as competitive inhibition, meaning that the molecule that inhibits the enzyme binds at the same active sites as the normal substrate for the enzyme. So here's the normal substrate for this particular enzyme, but here's this blue guy right here, which is a competitive inhibitor that also binds at the same active site of the enzyme. So that's why it's called competitive inhibition. They're both the substrate and the inhibitor. They're competing against each other for the same active site of the enzyme. Okay, except now if an inhibitor binds, is not the substrate, so it will block the normal substrate from binding at the active site. So here's a substrate coming in thinking that it can bind, but oh, there's a blue guy there inhibiting it, so it can't bind and bounces right off without catalyzing any reaction. So this inhibits the enzyme from doing its job. The other type of inhibition is called non-competitive inhibition, where the inhibitor does not bind to the active site of the enzyme, but a different site called the allosteric site. Okay, so now we're not dealing with competing over the active site. The inhibitor binds at the allosteric site, which is some other site other than your active site, and causes a conformational change in the enzyme such that the active site no longer fits the substrate perfectly and catalysis stops. This is also referred to as allosteric inhibition. Okay, and this is illustrated perfectly in this image here. Here's your enzyme right here in gray. Okay, and this is that active site for that particular substrate that is a half circle shape, for example. Well, originally this active site is perfect for binding to this substrate, but there is this allosteric site on the other side of the enzyme. If the inhibitor, non-competitive inhibitor, comes in and binds to that allosteric site, it causes a conformational change in the enzyme itself such that now the active site is altered and now no longer has this nice half circle shape, but have this weird shape. And the substrate coming in, oh, I can't bind there, I'm going to bounce off, because then this active site no longer is perfect for this substrate, thereby blocking the action of the enzyme itself via an allosteric inhibitor, okay, or non-competitive inhibitor. So how these two different types of inhibition affect enzyme rate or enzymatic rate can also differ. For a competitive inhibitor, because it binds to the same active site, it's competing against the substrate, it does not change the active site. Okay, so it will affect the initial rate of the enzyme reaction, but it will never change the maximal rate because the normal substrate and the competitive inhibitor are both competing at the same active site, and the substrate still does have a chance in binding to the active site. That's why the maximum rate is still the same as normal enzyme. Now, with non-competitive inhibitor, it changes the maximum rate of enzyme reaction because it literally changes the active site, so then it no longer binds to any of the substrates, okay? So it's not really out competing the substrate, but it changes the enzyme itself. So that's why for non-competitive inhibitor, it can actually lower your maximum reaction rate by quite a bit, okay? So that's what another difference between competitive inhibition and non-competitive inhibition. I know it's been an hour, but we only have a few slides more to go. So I would say maybe 20 more minutes. Okay, so bear with me. Let's finish this chapter up. So how does this apply to biology macroscopically? And this is some interesting stuff. Okay, so many toxins, poisons, antibiotics, and pesticides are actually examples of enzyme inhibitors. That's how they work. For example, pesticide is a good representative of an enzyme inhibitor where it's supposed to be specifically inhibits an enzyme called ACHE or acetylcholine esterase, which is an important enzyme for muscles to know when to relax. Now, when this enzyme doesn't function properly, your muscles end up continuously remaining in contraction and result in what we call rigid paralysis. Okay, you can't move. You're frozen into place because your muscles don't know when to relax. You have to have this enzyme in order for you to relax, which is why when you spray a pesticide on a pest, the insect kind of ends up freezing in place and then they can no longer fly or crawl because their muscles are frozen in place. Again, this will be something that we will talk about this enzyme immensely when we discuss the topic of muscle contraction in Anatomy and Physiology 1. All right, so the next example of enzyme inhibitor is antibiotic. Okay, antibiotic helps us to fight against bacterial infections, as we have learned in chapter 1 about penicillin. Remember, penicillin, we learned that was discovered by Alexander Fleming, is a molecule that was discovered in mold specifically, and it works by disrupting the production of bacterial cell wall, and it ends up being a lot easier for our immune system to destroy those bacteria via cell lysis when we have antibiotics around, okay? And last example, let me erase my thing there. Okay. And last example is something that we may not want to be exposed to are toxins, or poisons if you think about it. So a specific type of toxin called sarin acts kind of like what we're using in pesticide, except that it works for humans instead. It's like human pesticide, okay? But it is a potent inhibitor of ACHE in humans specifically. And this was actually released in Tokyo subway trains back in 1995 by cult members and killed about 12 people, and tens of thousands of people were severely injured. Tens of people were severely injured, and almost 1,000 of those people in the subway train end up with vision problems. Okay, so this is a very potent toxin that was released. Okay, sarin actually blocks muscles from relaxing, just like how pesticide does in insects, which results in victims being unable to control their bodily functions. That will result in twitching and jerking of the body. And then that will ultimately lead to suffocation and the victim dies. That's how the toxin works. But that's just another example of what enzyme inhibition is doing. Remember, I said a lot of our cellular reaction is not very efficient. It's not very quick. They can be spontaneous, but they may take a long time. So it always require the action of an enzyme. If we block those enzymes from doing their job, a lot of things shut off, and those things or reactions can be pertinent or important for our survival, that inhibiting that particular enzyme can end up killing us. Okay? All right, so I have mentioned that just as we can inhibit an enzyme, we can also act activate an enzyme. We call these allosteric activators. The fact that it begins with the word allosteric already tells you that the molecule will bind to a site that is different from the active site of the enzyme. That's the whole point of allosteric sites. So just that instead of causing the active site to become unable to bind to a substrate, allosteric activator actually makes the active site more attractive to the substrate. So here's, for example, here's the enzyme right here. Here's a activator, allosteric activator. It binds to an allosteric site. Its original active site is a gloop right here, which is not very, does not bind very well to your substrate, okay? But upon this activator binding, changes the active site to more of a spherical shape, then this curvature binds to the substrate perfectly, activating the enzyme. Now the substrate can go ahead and become whatever product it's supposed to become, and then the reaction finishes. Okay, so without this activator binding to the enzyme, this original substrate can never bind to the enzyme. And a lot of these allosteric activators come in the form of what we call cofactors and coenzymes. Now, cofactors are inorganic ions such as iron or magnesium or zinc. Okay, one example of this type of how the cofactor works is that DNA polymerase, which is an enzyme that is required for DNA replication before cell division, requires zinc ion in order for this enzyme to work. Okay, so that's why it's an allosteric activator. Without zinc, DNA polymerase doesn't do its job. While coenzymes are organic helpers molecules such as dietary vitamins. Okay, so you can tell that these are ions right here. That's why they're inorganic ions. Inorganic ions, cofactors. Organic molecules, coenzymes, and all of them are usually vitamins. So for example, vitamin C right here serves as a coenzyme for multiple enzymes that take part in the building of collagen fibers. Remember, we talked about collagen in our protein for chapter 3. And turns out that the process of making collagen requires enzyme that is activated by vitamin C. Okay, so make sure you understand the difference between cofactors and coenzymes. Okay, now, and for the application of our body system, sometimes we don't want to constantly activate an enzyme to continuously make the same product. No, if we have enough of what we want to make, we better stop to not waste the resources or even overwhelm the system. So this can be done in what we call feedback inhibition, meaning that if we a certain pathway involving many enzymes, for example, as you can see here, enzyme 1, enzyme 2, enzyme 3, so this pathway involves many enzymes in succession to produce a final product at the end here. Sometimes this particular end product can actually serve as an allosteric inhibitor of the very first enzyme of the pathway to stop the assembly production line. Okay, so meaning that if we have a lot of that product, we need some way to tell that enzyme, hey, you better stop working because we don't need a lot of us anymore. We have a lot of our products. So that's why the product end up being an allosteric inhibitor, changing the active site. The substrate no longer binds and the production assembly line stops. Okay? So that's a way of feedback inhibition. And when a product specifically inhibits its own production, we call that a negative feedback. Again, this will become another major concept if you need to take anatomy and physiology, because our body system are working in a way that consistently utilizes these different feedback mechanisms to maintain homeostasis for our body, like we have introduced in chapter 1, what homeostasis means. And as a wrap up of this chapter and introduction to the next chapter we will be discussing, which is chapter 4 on cells, In eukaryotic cells, enzymes and molecules are actually compartmentalized into different organelles. For example, enzymes required for certain cellular processes are housed separately for more efficient chemical reactions. Okay, for example, enzymes involved in cellular respiration. Okay, the latter stages of cellular respiration takes place exclusively within the mitochondria right here. Okay, you wouldn't see enzymes for cellular respiration in the nucleus membrane or the rough ER, because that's not where all those all those things we need are. Okay, all those things we need for cellular respiration, the latter stages anyway, is within the mitochondria. So might as well have those enzymes housed within the mitochondria. And on the same line, enzymes involved with digesting cellular debris and foreign materials are located in lysosomes. Okay, for example, these little spherical molecules there. We won't have enzymes for breaking down things in the nucleus or on the rough ER, or else we'll be in trouble. We'll be breaking down things we're not supposed to be breaking down, okay? Only broken stuff that is supposed to be broken down will go to lysosome, and lysosome is where those enzymes specific for breaking down things are housed. Okay, so things are housed around the cell and compartmentalized in their particular locations, because those are the enzymes for those particular reactions that is necessary for that particular area. Okay, we can't put them into the wrong area. Okay, so that's all we have for chapter 6, okay? And this whole organelle and their function stuff is something we'll talk about in chapter 4 next. The functions of the different organelles and compartments of bacteria, animal, and plant cells, okay? So I will see you again in chapter 4.